Hi, my name is Gillian Antunes and I'm a postdoctoral research associate at the Computer Science Department of Rice University. This is the second part of a video tutorial on HLA-Arena. You can get started by checking the GitHub repository for HLA-Arena, where you can find some basic information on how to install Docker and how to pull the HLA-Arena Docker image from Docker Hub. So you can go ahead and copy this command here and just execute in the terminal in any system that has Docker installed. You can just do that into a terminal. In my case, I have recently downloaded the image, so there is not much to be added. It was quick. If you're doing this for the first time, it will take a few more minutes. The whole image has about 14 gigabytes that need to be downloaded, but the good thing is that Docker controls this download, and even if you have some interruptions, you can easily complete the task. So once I download the image, the next command here, kind of long command, is to copy the workflows to the folder you are in. So you can either do this in the folder you are, or you can maybe create a specific directory for running these experiments. So I'll create one folder here, just for this case, and then I will run this command. You can just copy and paste, and then you'll be able to copy the files, the workflows that we'll be using in this demonstration. Okay, so the next command will be to launch the container and share the folder. You can see that all those files were copied to the folder I am in, and now I will launch the container to analyze these files. So now I'm executing workflow. I execute the command to run the container, and you can see you have some links here. You can just copy and paste in your browser or control click, and then you see the interface of the Jupyter notebooks. And if you click, for instance, here, each one of these files that has this extension here is one of the workflows available, and here's some data that some of these workflows are using. So if you click in the number 0 here is a minimal example. So it's a very simple workflow that allows you to run the steps for modeling a peptide-HLA complex using ApeGen, which is one of our applications. So in the top you have all the commands of the Jupyter notebook that allow you, for instance, to add cells or to remove cells from this notebook, and you can learn more about in the specific documentation. But basically here you have different cells that can contain either comments, you can see highlighted in blue, or code highlighted in green. And in fact, you can see here how this cell is defined. And in both cases, you can double click the cell to edit the content here, and then you can execute the cell clicking here, or you can also execute the cell by holding Shift and Enter. In this case, it was markdown content, so you see that only formatted the text. In this case here, I have some code that will import the HLA-Arena library and will create a folder in which we'll run this workflow 0. So if I run this cell, you can see it's running by the asterisk here. Now it's completed. It created this workflow 0 inside experiment 1. So you see inside the container, the shared folder is called data, but this data folder inside the container is shared with this experiment 1 folder in my local computer. And you can see that it created this workflow 0 folder that is here, and here you have the workflows that we are using, and right now I'm executing workflow 0. And note that the container has root permissions over the shared folder. So everything that's created by the container inside this folder will be protected from your user. So later on, if you want to edit these files, you'll have to change the permissions of this folder and its content. I will leave this folder open on the side so that we can see the files being created. And then I will proceed to the next step, which is obtaining the receptor for docking. In this example, we'll just obtain an experimentally determined structure for HLA-A2402, and this structure can be obtained directly from the protein data bank by just typing the PDB ID here. And there is a function that will pull this structure and also do some basic processing with PDB fixer, so that's ready for docking. You can see here the structure was downloaded, and now this HLA allele variable refers to that structure. There is some additional documentation that we'll come back to it later, but I'll move on to the next step, which is actually running the peptide docking with ApeGen that I have discussed in the previous tutorial. So you can see here that we are defining the sequence of the peptide as input, and then we are using the structure that we selected before. And this is the input, and the output is being processed here. You can see that the sequence of the peptide, the receptor, ApeGen go on with the alignment of the both termini of the peptide, and then it fills this gap with the random coordinate descent algorithm. And after that, it goes on with the reconstruction of the side chains using SMINA and running this local optimization of the side chains of the peptide and the HLA. This whole process takes about 2 minutes, but I'll just skip ahead for sake of time. You can see here that ApeGen has selected a number of conformations, and then we are executing an extra step here, that is for each of these conformations we are running a global optimization with OpenMM, and here you have the lowest energy conformation obtained in this process. All these files are saved here, so you can see this is the lowest energy minimization structure that you can analyze, and here you have all the other conformations including both the peptide and the receptor. But in fact, you can visualize the structure without leaving the notebook using NGL Viewer. As you can see here in these 2 cells, first we are having a cartoon representation of the peptide in the binding site, and then there are different ways to visualize. You can also change the code here to show, for instance, surface representation or how other ways. And then here you have a representation of the entire ensemble of conformations that was created, only seeing the peptides, but as I mentioned, you have the peptides and the receptors saved in a folder. And then these files are available for different analyses that you might want to do subsequently. This concludes this first example of how to run HLA-Arena to use ApeGen and model peptide-HLA complex using the crystal structure of the receptor. As I mentioned, there are other workflows, and you can see in the documentation and in the other workflows how to customize these commands and make this fit your needs. So, for instance, you don't need to download a structure to use as the receptor for docking. The part that I skipped here mentioned that you can actually model the HLA receptor using Modeler and using homology modeling, but for that you need to first set a modeler key, and the information about that is provided in the documentation. So you can register in the modeler web3 page, and then you need to run these commands once to set up this modeler key in your HLA-Arena container. Once you do that, you can go back and execute this workflow in the way that you'll use Modeler. I have already registered my Modeler key, so I'll go ahead and demonstrate how to use it. First, I will clean this folder here and to start over. And I will now comment out this content because I don't want to download this PDB structure anymore. So I'll just change the cell to Markdown so that is not executed as code. And then I will bring this cell here to work as code. But then instead of 2402, let's model 0101. And I want to model a peptide that is a melanoma-derived peptide from AJ3. And then let's see the results. So I can just go back and execute everything now. So let's execute and see what happens. So now it's creating the, repeating the steps of creating the folder, and then here you see it started the modeling process. And what happens is that HLA-Arena will use the HLA allele name provided and will fetch the proper sequence from the IMGT-HLA database. And it will also look for a valid structural template from the protein databank based on the supertype classification. So in this case, it found the 3B08 structure, which is a structure of A0101 receptor. It downloads and makes a pre-processing of this receptor, so that's good for the homology modeling, and it goes on modeling this receptor. So this will work for most classical class 1 HLA alleles, HLA-A's, B, and C. It will not work for the non-classical HLA alleles, and it will not work for class 2 HLAs, but you can also change this code so that it uses your specific HLA sequence and a given template that you choose. So in that case, you can also model other class 1 alleles and even MHCs from other species. It will not be as easy to run these for class 2 though, because ApeGen is not currently set to work with class 2. So a few minutes later you have the modeling results, and then a low energy conformation is selected for docking, and you have the same execution of ApeGen as before, but now using the model structure. And then you go back to the visualization with NGL Viewer, and you have here the final ensemble of conformations the same way as before, and you have inside the folder all the conformations of the complexes for further analysis. I hope this tutorial was able to give you a basic understanding on how to use HLA-Arena and how to execute the different workflows. Please do check the documentation to see the additional functions available and the additional workflows. You can use the workflows as they are, or you can start combining commands or cells from different workflows and building your own pipeline or workflow. We also have additional information and experiments describing the use of these workflows on our manuscript that was published on JCO Clinical Cancer Informatics. And in fact, you can use some of these workflows here to reproduce the results presented in the manuscript. So for instance, I will quickly execute this workflow through 3B. In this case, we are conducting a large-scale virtual screening of peptides. So we are analyzing 9,000 different peptides bound to 6 different HLA alleles, and we are combining sequence-based. We are doing the homology modeling of these HLAs. We are using sequence-based prediction to filter some of the peptides with MHC flurry, and then you can define a threshold here of how many peptides you want to select for the structure-based analysis, and you do the peptide docking with ApeGen, and then you can do a structure-based ranking of these conformations based on the scoring they get. And this dataset was on purpose composed of a number of known binders, experimentally determined binders for each one of these alleles, and a number of decoys that are not binders. And then you can see that this combined analysis with sequence and structural ranking can enrich the bottom of the distribution here with true binders. So this is what you expect if you want to use this for real structure-based virtual screening or this combined sequence and structure-based virtual screening of HLA binders for different HLAs. And actually, the structural analysis can compensate for some of the limitations of the sequence-based prediction, especially for less prevalent HLA alleles. So please go ahead, play with our different workflows, create your own, and don't forget to cite us. Thank you very much.